All right, I'm going to try talking a lot faster because my last two YouTube videos have not been processed. So here's hormones. They are small organic molecules which humans and all organisms have that are multicellular. They coordinate development and timing of things, and they relay messages deep into the tissues in multicellular organisms. They are produced in one set of tissues and then migrate to another tissue for an effect. In us, it's our bloodstream. In plants, it's xylem or phloem. They work in a dose-dependent manner or concentration-dependent. They work in very tiny concentrations, but under a small window. So don't take too much of a hormone. It can have negative effects, like too much estrogen can lead to some forms of breast cancer in women. In plants, they're a little more diffuse. They are produced in one tissue, move to where their target cell is. That target cell will have a receptor where just that hormone binds in a lock and key fit. Auxins were the first of the discovered. They were accidentally discovered by Darwin, whose plants on his ship would lead towards his candle light. Now we know it was the blue color from the candle that was binding or activating the cryptochrome photoreceptor or the phototropin blue light receptor. It is responsible for delaying leaf dropping when it is produced in leaf blades. It causes apical dominance, which is the top of the plant grows the best, like a Christmas tree. And lastly, it can cause roots in soil and in tissue culture. There are many different compounds called auxins. The one that occurs naturally is IAA, indole acetic acid. They're made from tryptophan, which is a amino acid. It causes mainly acid growth through what's called the acid growth hypothesis. If you put cut segments of a seedling into water, they do not stretch, whereas if you put them in water with auxin, they do stretch, and the previous slide showed a typical dose response curve. They are produced in the top of the plant, and then they migrate down the plant. And so they're made in the top of the plant, they move down and prevent lateral buds from growing. If you remove the top of the plant, like a horse or a cow, or you prune a tree back, there's no more auxin, so the lateral buds begin growing. You can see that in this middle plant. If you trick the plant into thinking there's still auxin by applying it to the tip of the decapitated plant, once again, lateral buds do not grow. The acid growth hypothesis is attached as a PowerPoint, but basically acid pumps get involved and they pump acid into the wall, which activates enzymes called expansins. They loosen the cross linkages between cellulose and the rest of the wall structures, and the trigger pressure then causes the cell to expand. Gibs. Lots of gardeners know about gibberellic acid. They are a large category of many different compounds. We just put a number behind the GA. There are more than 135 of them. They're made from the mevalonic acid pathway. They cause normal plants to grow tall, whereas dwarf plants either can't make GA or they make GA, but the receptors are broken. So you can spray gibberellic acid onto a plant. It will grow tall if it was a synthesis mutant. It will stay short if it was a receptor mutant. It triggers germination by releasing amylase. It will also cause flowering if you have a very mild winter. Here is little Marvel pea plants in the lab that have been sprayed with jib. They grow tall on the right, so that was a GA production mutant. They also stretch out the stems for Thompson seedless grapes, the one on the right. They cause bolting in what are called rosette plants like mustard. You can see on the left, that was just a watered mustard. The one on the right has been given higher and higher levels of jib. It causes stem hyperelongation and then flowering. It during seed germination, the allurone layer of grain cereals releases amylase. The amylase converts starch to sugar, and the embryo then grows. On the right-hand side, 4 seeds were placed on a dish that had gibberellic acid pretreatment. The halo here, the clear area, shows where amylase has been produced. Much more with gibberellic acid than on the left without gibberellic acid. Sorry, I'm trying to go fast. Cytokinins sound like the word cytokinesis, cell division. It is made from the mevalonic acid pathway. It is made in the bottom of the plant and moves upwards, called acropedal. It is high in coconut milk because we believe it helps the coconut float to an island and initiate germination and cell division when it lands on a sandy beach. It can release lateral buds from apical dominance even if auxin is being produced in the tip of the plant. It's also a fountain of youth. It prevents aging and death or senescence in wherever you apply it onto your plant. Once again, it causes shoots in tissue culture. Auxins, remember, favor roots, and if they're equal, you produce undifferentiated parenchyma cells called callus. On the left, you can see this drop of cytokinin caused this lateral bud to grow that whole length, even though there was still the tip of the plant present. The control is on the right. It was applied here on the left. You can see it delays death. It remains green. On the right, ethylene has caused death and aging. That was ethylene. On the right, cytokinin on the left. ABA and ethylene are smashed into one chapter. ABA is one chemical made by the mevalonic acid pathway. It prevents early seed germination, so it imposes dormancy. It also helps dry out the seed or desiccate the seed. It also closes stomata during water stress. This shows once again the mechanism of ABA on the right, freezing the proton pumps so the stomata close. This is what happens in a mutant of corn on the cob that cannot make ABA. You see all the seeds have begun sprouting even before they're mature. Ethylene is a small molecule. It's also produced by burning, so trees around burning gas lamps can lose all their leaves. Also, if you drop a fruit, it will wound the plant, and that will lead to ripening. It's made by mevalonic acid. It causes ripening of climacteric fruit. Not all fruits behave under ethylene control, but ethylene, when it is active, it will upregulate transcription and translation of genes like cellulase, for instance. This is degrading cellulose, softens the walls, and that's why your fruit gets softer. Ethylene can also activate Ethylene can also activate cellulase in the abscission zone where the leaf connects to a stem. And so in the fall, when leaves drop or abscise, that's because cellulase has nibbled the cell walls in the abscission zone. Lastly, ethylene causes epinasty, a downward folding of leaves on some plants. You can see this tomato plant in this bell jar with a ripe, rotting banana is showing epinasty, downward folding of leaves. You can try to block ethylene by adding silver to cut flowers. Silver thiosulfate, after 2 weeks, they look much fresher because the ethylene is produced, but the receptor is blocked, so the plants do not show petal senescence like they do in the water-treated control. Here's a summary table, and there's also another table attached in your document.